A very good morning to one and all present here. It gives me immense pleasure to extend a cheerful welcome to you all. On behalf of the Department of Physics and Chemistry, Sona College of Arts and Science, I am very delighted to welcome you all to the webinar on Nanomaterials for Environmental Remediation by our Honorable Chief Guest, Dr. R. Parimala Devi Ma'am, Assistant Professor, Department of Physics from Mother Teresa University, Kodaikanal. I would like to thank our Sona management, Principal Sir Dr. Kader Nawaz, and the department for providing us with a platform to learn new things. Now, I invite Dr. Nilufer Ma'am to introduce our speaker of the day.
Okay. Okay, good morning to all. In this webinar, now I am going to discuss about how to detect and degrade the environmental pollutant. So this, we know there are different types of pollution are there. That is noise pollution, air pollution, soil pollution, water pollution, like that. It is very yes.
Ma'am, slide is not changed.
Yes, I didn't.
Yes. Okay.
Okay, so there are different types of pollution, and all that pollution are very hazardous to our environment, and it is very dangerous to our human beings also. So in this context, the World Health Organization also reported that over 3 million people die each year from the effect of air pollution. In addition to that, World Energy Congress suggests that if the world continues to use fuels reserves at the current rate, then the environmental pollution in 2025 will create irreversible environmental damage. So what is the cause of this pollution? It is because of our ongoing industrialization and urbanization. It releases number of toxic chemicals into the environment. So it pollute rivers, soils, and everything. And this source of contamination in water include the process related to municipal, industry, and agriculture waste, and it will create waterborne bacteria and some organic pollutant that enter into the water and soil, and it is very hazardous to our environment. So we need to remediate that pollution because pure water, it become very limited short resource nowadays, and many pollutants in water streams have been identified as a toxic and harmful to the environment and human health. Because of its extreme toxicity, the detection and removal of organic pollutant from the soil and water demands priority in pollution control effect. So in this, so we have many, traditionally we have many methods to remove the water pollutant. Mainly in this webinar, we're going to discuss about how to detect and degrade water pollutant and soil pollutant. Okay, and there are many methods in traditional, like boiling, like that and all, but now it is now we are using nanomaterials to detect and degrade this pollutant. Okay, why we are preferring now nanomaterials is because the revolutionary progress in the area of nanoscience and nanotechnology over the last couple of decades has motivated the scientific community to explore this high potential area of research where the unique advantage properties of novel nanostructure material could be exploited to deliver more for the prevailing water related problems. Normally nanomaterial, it have a dimension from 1 to 100 nanometer. Because of their small size, they contain quite lesser number of atoms, which in turn give rise to very different properties in them as compared to that of bulk material. Owing to their small size, nanomaterial possess very high surface to volume ratio, which leads to more surface dependent property. Okay, so this has been found that due to their advantage physiochemical property, nanomaterial are excellent absorbent for variety of water pollutant. Nowadays there are several types of nanomaterial is used for water remediation, like metal nanoparticle, polymer nanoparticle, zeolites, carbon-based nanomaterials, biopolymers, ion nanomaterial, and nanoscale semiconductor photocatalyst. So there are a number of materials which we number of nanomaterials now we are using for remediation of remediation of environmental pollution. So first in water, we have organic pollutant and also some bacteria in that. First we want to detect or degrade that pollutant. For that also, because water infected with that pathogenic microbes is lethal to human health. As per World Health Organization, annually about 3.4 million people die as a result of various waterborne diseases around the world. So this normally we use antibiotics for this, but prolonged use of antibiotic has allowed the microbes to undergo genetic mutation and develop resistance against them. Such evolution of antibiotic resistant microbes is serious threat to all living beings. As an alternative of some traditional techniques, disinfection recently nanomaterials certain nanomaterials such as metal nanoparticles are used for against microbes and also to control that. So for large number of nanostructure material have antimicrobial property has been synthesized and used for microbial control. Among them, silver, titanium oxide, zinc oxide and carbon-based nanomaterial are most extensively studied nanostructure antimicrobial agent. Since long time, silver is known for its antibacterial and sensing property. But when used in nano form rather than bulk, its activity get enhanced. Okay, several nanoparticles exhibit strong broad spectrum antimicrobial activity. It kills both gram positive and gram negative bacteria. Now most importantly, it has very less toxicity towards human cell and it is easy to use. This makes nano silver a most popular material for wastewater treatment. Normally this we now came across the number of advertisement like Spain's and number of soaps they are using nano silver or activated carbon nanomaterial for antibacterial activity. Likewise now in commercial application also many water purifiers like Tata Schwach, Blue Star, Aquapura system etc. they are using nanomaterials. Okay, so actually what is the mechanism of this antimicrobial activity is shown here. This is a nano silver. This silver it's taught when it reacts with this bacteria, it readily bind with the thiol group of some crucial proteins. Okay, it affect the proteins of the bacteria and it also damage the enzymes. And this silver it also blocks the DNA replication a process and it cleaves the cell membrane. So this nanostructure silver or whatever semiconductor, that is zinc oxide, titanium oxide, they will generate ROS, that is nothing but ROS is nothing but reactive oxidation species. So it release this and it normally it will release inside the cell, which this ROS it normally which it helps to killing the microbe via membrane cleavage and protein damage. So this silver it effectively acts antibacterial agent. Okay, so but this is very costly. Silver and gold, this noble metal is very costly, so we go for some other material. For that we worked for that and we produce some copper nickel material for that act against bacteria. So that work was published recently in this materials letter. That work is nothing a synergetic effect of copper nickel bimetallic nanoparticle and nanoparticle for enhanced bacterial inhibition. So in this, actually, we know most of the metal nanoparticle have been studied extensively as bacterial growth inhibitor due to their strong antibacterial property, stemming from a smaller particle size, electrostatic interaction originating from the surface charge, and production of reactive oxygen species on the nanoparticle surface. Okay, here copper so that silver and that gold is very costly, so we go for copper nanoparticle. We try to prepare copper nanoparticle, which is also a strong and which also shows strong antibacterial activity against a wide range of bacterium, owing to the bacterial cell wall disruption and distortion of helical DNA structure. This copper based bimetallic system enhance the inhibitory activity of metallic nanoparticle. So it also combined with silver and gold, and that was widely studied. But here, even silver and gold, we know it is very costly, so we go for economical bacterial agents. So we prepare copper nickel bimetallic. Okay, that was prepared using the solvothermal method. Solvothermal method is a simple method you can easily prepare if you have this autoclave and a furnace. Okay, for that we take appropriate amount of copper sulfate and nickel nitrate, and it was add with sodium hydroxide. So and here we use hydrogen. This hydrogen, it acts as a reducing agent. When you add this hydrogen, this copper sulfate will reduce into copper and nickel nitrate will reduce into nickel. Okay, this was mixed and stirred for 2 hours. Then it and for this copper sulfate and nickel nitrate we use ethylene glycol as a solvent. Okay, so because of the solvent, this method we call it as solvothermal, and it is heating, so we call this method as solvothermal. We transfer this with Teflon coated autoclave, and we transfer this Teflon coated autoclave to furnace, and it was treated at 150 degrees Celsius for 5 hours. After that we get a black precipitate. That black precipitate was washed repeatedly with distilled water and then with ethanol. Then we got a copper nickel nanoparticles. Okay, this is the this copper nickel nanoparticle, then it is subjected to different characterization to study its structure, morphology, and also we also analyze its antibacterial activity. Okay, first we go for powder diffraction method. In powder diffraction method we can analyze its structure. So using this from this pattern we clearly known that copper, it is a face center cubic structure, and also nickel also face center cubic structure. Then this copper nickel, it also shows a same face center cubic structure, but there is a slight variation or slight shift in the peak position when compared to monometallic. There is a slight shift in bimetallic. Okay, so this was due to of copper atom in nickel matrix and subsequently partial replacement with nickel atom. Okay, we using D by Scherer's formula, we also find the crystalline size of this particle, monometallic and dimetallic particle. Here this shows copper shows 25 nanometer and nickel shows 25 nanometer, and copper nickel bimetallic is 23 nanometer. Okay, so then it is a FTA or spectra of this bimetallic nanoparticle. Normally here the peaks observed at 640 to 620, it is due to the metal oxygen vibration. The peaks at here 3450, here this is due to the OH stretching, and peak here 1571, 1420 and 1117 is due to C double bond CH and CO, that is due to the precursor which we use to synthesis. Okay, that also available in this FDR spectra. And next we studied morphology. Okay, we studied the morphology of this copper nickel and copper nickel bimetallic using SEM analysis. SEM is nothing but scanning electron microscopic analysis. Using that we can study the morphology. So this picture shows copper, it is a spherical cuboid structure, and nickel, it is a spherical structure with spikes. And when we synthesis copper nickel, it shows a porous flaky interlaced with a spherical structure. This is an EDAX spectrum. EDAX is nothing but energy dispersive and X-ray analysis. It shows whether it shows whether the compound is present or the mass of the compound. Okay, from this EDAX spectrum we identified copper is present in this and nickel, and both bimetallic is present in this copper nickel. Okay, copper nickel nanoparticle. Okay, next is important here that we are going to see the antibacterial activity. This was studied using diffusion technique, agar diffusion technique. Normally in this we take an agar and we pour a bacterium and we also add our drug. Okay, we compare our nanoparticle with a standard drug, cefaxamine, that is a third generation drug normally used for pneumonia and all. And so here we take our nanoparticle also as a drug and we analyze. This is for this is for gram positive. Okay, this is for gram positive bacteria. This is against gram positive bacteria. So Streptococcus aureus, this act as this we taken as a source here, and we studied antibacterial activity. Here the zone of we can see the zone of inhibition. So this is for 30 millimeter for copper and nickel nanoparticle, and zone of inhibition for copper nickel nanoparticle is 31 millimeter. And we also analyze antibacterial study against gram negative, that is E. coli. So this is also very dangerous bacteria only. So we studied against this and we saw the inhibition zone of copper nickel particle is more than that of copper and nickel. So this copper nickel nanoparticle, it acts as an antibacterial agent. So normally the higher we saw the higher zone of inhibition was recorded for bimetallic than monometallic. It is due to the unique distribution of two different metals, thus enabling the release of two different metal ions. Copper, copper 2 plus, normally this copper, it gives copper 2 plus, that binds with the DNA and it destroys the helical structure of DNA. And this nickel, it helps to displacing zinc from the secondary binding site of metaprotein. Okay, so this is very helpful as an antibacterial agent. So normally this nanoparticle, it damaged the bacterial cell membrane and it disturbed the enzyme function. It also alter the local pH. It also produce more reactive oxygen species. Normally it will kills the bacteria. Thus this copper nickel nanoparticle, it act as a very good antibacterial agent. Okay, so so the infected, that is normally the wastewater, it consists of many bacteria and virus that leads to many diseases that can be degraded using the using that nanoparticle. Next is photocatalytic degradation. Photocatalytic degradation, it is normally we are in Salem and Erode, and normally normally in Erode and Salem we have more dye manufacturing company, textile industries. Okay, so this dye manufacturing and textile industries are chief contributors to the problem of water pollution. Normally the textile pigmentation and treatment process, it contribute 70 to 20% of global industrial water pollution. Okay, so annually 7 into 10 to the power of 5 tons of dyestuffs are produced worldwide, and it normally used to release in our environment during synthesis. Okay, so the release of such untreated waste into the water bodies is resulting in destruction of aquatic life and disturbance of the ecological balance. It also lead to various disease and health problem in human beings. Degradation of such water pollutant using light absorbing material under solar irradiation is the economic and highly efficient way of wastewater treatment. Though the photocatalyst is easy way to get rid of this harmful organic dye present in wastewater. Okay, normally the photocatalysis, it is a process of accelerating a light induced reaction in presence of suitable heterogeneous catalysis. Normally this heterogeneous catalysis is a nanoparticle. Normally semiconductor nanoparticle act as a heterogeneous catalyst. Okay, now we know the semiconductor, it have a wide band gap between the conduction band and valency band. So that there we have no there will be no energy level that won't avail the no energy level is available at energy gap to promote recombination of electron hole produced by the photoactivation. When photon with energy equal to or greater than the material's band gap is absorbed by the semiconductor, an electron is excited from the valency band to the conduction band. So it will generate normally photo generated electron hole pair that normally we called it as exciton. That excited electron and hole can recombine and release the energy gained excitation of electron as a heat. Excitation lead to inefficient photocatalyst. So our ultimate goal of photocatalyst designed to facilitate reaction between excited electron with oxidant to produce a reduced product, and or a reaction between a generated hole with reductants to produce a oxidation product. Due to this generation. Here normally the photocatalysis we use a semiconductor, normally as a photocatalyst we use semiconductor nanoparticle. Photocatalysis is nothing but light, that is the reaction is induced by light, that we call it as photocatalyst. Using that photocatalyst we can easily degrade the pollutant in water, okay, because our semiconductor it have a band gap that will when the light is absorbed by that semiconductor material, it normally produce electrons and holes, and we should reduce that recombination. So that is the mechanism in this photocatalyst. If you reduce that recombination, it will easily degrade that organic pollutant. Here normally when this holes and electron it undergoes with a pollutant, it will produce OH. So normally that dye, it is an biodegradable and a toxic material. When we add a photocatalyst, it will produce this OH material. Okay, OH. This is easily degradable. This hydroxyl product, it is easily degradable. So that using photocatalyst, we can convert a non-degradable product into degradable product, so we can easily remove or degrade that organic pollutant, okay, using this mechanism. So to understand well about this photocatalysis, now we can discuss about another work, that is synthesis, characterization and photocatalytic activity of fluorine doped TiO2 nanoflux synthesis using solid state reaction method. Okay, so normally here we use titanium oxide as a photocatalyst. Titanium oxide is titanium oxide. This is very it is one of the functional semiconductor metal oxide that performs high efficiency in photocatalytic activity because of non-toxicity, excellent energy conversion efficiency and long-term chemical stability. Normally it have higher range of band gap, so we want to reduce that band gap by some doping, okay, so then only our photocatalytic activity will become more efficient to remove the waste, to remove the pollutant. So for that we dope this titanium oxide with the fluorine. Titanium oxide with the fluorine. Normally fluorine doping in titanium oxide converts Ti4 plus to Ti3 plus by charge conversation, and existence of 3I3 plus can reduce electron hole recombination rate. So subsequently it will enhance the photocatalytic activity. So fluorine doping may enhance the visible driven photocatalyte due to the creation of oxygen vacancies. To synthesis we choose solid state synthesis method. It is very easy method, you can do in your lab also. That is just take titanium oxide and ammonium fluoride in appropriate amount, and you just take that product mixed well with mortar and pestle, and you transfer that metal into the crucible, and you treat that material in muffle furnace at that it is calcinated at 500 degrees Celsius for 5 hours. Then you get an yellow color powder. You just grind that and you wash that and you can subject it to different characterization. Okay, so first we go for XRD. XRD is nothing but X-ray diffraction, X-ray diffraction pattern. This shows the structure of titanium oxide. So this is from this we can identify the structure of titanium oxide and fluorine dope. There is not much change in XRD because both this oxygen and fluorine it have same radius, atomic radius. So when the fluorine occupies the oxygen space, there will be no change in that interfacial pattern. So there is not much change in this XRD, and here it shows a morphological pattern. This was studied using as scanning electron microscope. This image shows the morphology of the prepared sample. Thus it shows nanoflake structure. So next we go for optical absorption study, that is done by UV. Here TiO2 it exhibit absorption band at 420 nanometer, and when we introduce a fluorine in TiO2, here the absorption was shifted towards red end. So from using this graph, we modify the graph using this Monk model, and we plot a tough plot here. Okay, so this using this tough plot we can find the band gap energy of this titanium oxide and fluorine dope titanium oxide. So the band gap of fluorine was 3.09 electron volt, and fluorine dope TiO2 shows the band gap of 2.85 electron volt. So here the narrow band gap get reduced, that is the band gap it become narrow. So the reduction in band gap denotes the localized nature of fluorine spectra in TiO2 lattice. This is normally contribute due to the creation of surface oxygen vacancies that may reduce the band gap. And next is FTIR spectrum. This is fluorine transform infrared spectroscopy. It is normally used to find the band structure of TiO2, and here we observe a strong band here at 3000. This is due to OH stretching, and this 1500 it is also water. This is also due to water, and here this band it is due to metal oxide doping. So when you see this this have more hydroxyl absorption. This hydroxyl group on particle surface this is here it is more this OH it absorb more than that of pure TiO2. So this hydroxyl group on particle surface contributes to the improvement of photocatalytic activity by their interactions with photogenerated hole, giving to the better charge transfer which inhibits recombination electron hole pair. Okay, so next is this is an important study, that is photocatalytic study. Here for that we choose a methyl orange. Okay, methyl orange it is a hazardous dye. Normally it will be used in our textile industry. So now we want to degrade this methyl orange using this titanium oxide. Okay, using this nano titanium oxide. Okay, for that first we go for first we study here. This is absorption study of pure dye, and after adding TiO2 there was reduction in this absorption band. Okay, there is decrease in intensity. So this shows that is by adding titanium oxide the dye it get degraded. Okay, this was and here this is due to the fluorine dope TiO2. Here the intensity is very less than that of pure TiO2. Okay, so this fluorine dope it again improve the degradation efficiency. Okay, fluorine doped TiO2 it have greater degradation efficiency than TiO2. So this degrade the dye. Okay, we take just a dye and degrade using this TiO2. The nanoparticle it start to degrade this dye, and using the concentration at the concentration of initial concentration and the concentration at after this is done for every 10 minutes. So using this, that is using that we draw a plot between time and concentration. So this is This is rate constant slope. Using this, we can identify the rate constant value. Normally, the fluorine doped value have this is rate constant value, and this is very much rate of decomposition is normally it will be higher for that fluorine top TiO2 than TiO2. So this synthesis to product, it start to degrade our dye. So this so from this we can identify that this fluorine doped TiO2 used to degrade the dye product that available in textile industries. Okay, so this is the summary of that second work that we confirms that fluorine doped. It shows higher photocatalytic activity than pure titanium oxide nanoflags, which could be attributed to smaller crystalline size, well crystalline phase, band gap, and intense absorption in visible light region. So next we are going to, this is an important work, we are going to detect the organic pollutant. Okay, in water. Water, it consists of many pollutant that dye, everything, it is an organic compound, and that organic pollutant we are going to degrade. This is very hazardous. That normally persist in water, so that we call it as persistent organic pollutant, that are class of highly hazardous chemical pollutant that are recognized as serious global threat to human health and to ecosystem. So some of that persistent organic pollutant are dichloro-diphenyl, trichloroethane, and polychlorobenzene and dioxin, etc. These are very hazardous pollutant in environment. It is available in all the regions of the world. So we want to detect that pollutant, and we also want to degrade that pollutant. Okay, so so normally every person, it carries a body burden of that persistent organic pollutant, mainly in his or her fatty tissues, and they have a half life in soil, sediments, air, and more hard to biodegrade. In recent year, great research interest has focused on detection and degradation of this pollutant, in which techniques that are able to detect this compound is a trace amount or essential. This is because uh, this is because they can accumulate in human bodies through the food chain and cause severe disease when the concentration exceeds a critical level, even when just in trace amount in the environment. So the detection method of that pollutant is very important. So before 2 decades, they normally use gas chromatography or mass spectroscopy method, combined gas chromatography mass spectroscopy method, but that is expensive and it is also time consuming. So and it also not able to distinguish all the chemicals present in that. So we go for another important technique, that is surface enhanced Raman spectroscopy. This is an effective way to detect organic pollutant. Okay, now this surface enhanced Raman spectroscopy is same as our Raman scattering. That is, Raman scattering is nothing but inelastic scattering of photon by matter, meaning that there is an exchange of energy and change in the light acceleration. When we introduce a light, it start to it will start to the vibrate, the atoms in the compound, it start to vibrate, and that vibration will be sometimes equal to that light frequency, or sometimes it is greater than that of light frequency. So that we call it as Raman scattering. And when you use a nanomaterial, when the light is absorbed by nanomaterial, that vibration, it will be enhanced. Okay, so Raman signal, it get enhanced, and due to that Raman signal, we can easily find what are the molecules, even a single molecule present in that compound. That technique, we call it a surface enhanced Raman spectroscopy. This has attracted enormous attention due to its excellent performance and potential application in the detection of molecule in trace amount, even a single molecule detection. So this is mainly based on electromagnetic enhancement induced by a nanostructure noble metal surface and associated to the localized surface plasmon resonance. This is nothing but when you interact light with a noble metal surface. Noble metal is nothing but gold, silver, platinum, copper, like that. When you introduced light, that is electromagnetic signal, in the metal surface, it start to vibrate, and it produces a frequency that we called it as local. That frequency is equal to that of electromagnetic induction, so that we call that oscillation, it produce some resonance frequency that we called it as localized surface plasmon resonance. So normally this localized surface plasmon resonance will enhanced. Okay, so this is highly depend upon the interaction between absorbed molecule and the surface of plasmonic nanostructure. Absorbed molecule is nothing but here it is an organic pollutant. Okay, that is the this SERS, it mainly depend upon that organic absorption of organic pollutant and our plasmonic nanostructure. Plasmonic nanostructure is nothing but silver, nano silver or nano gold, like that. So SARS can be used to detect some organic compound even at trace level, okay, using our noble metals such as copper, silver, gold as a substrate. Okay, using so in this context we have an idea to synthesis a gold nanoparticle, and we form a nanocomposite with a semiconductor titanium oxide. Okay, then we studied we deduct the organic pollutant using that gold silver nanocomposite, and we also try to degrade that organic pollutant using same. So this gold titanium oxide nanocomposite, it has a dual function, that it will detect the pollutant and also it degrade the pollutant. Okay, so this work was published in Journal of Cluster Science, and for that first we synthesis gold using chloric acid. Okay, using chloric acid. Here the appropriate amount of chloric acid, that is here 4, that is tetrachloric acid, and we take appropriate amount of chloric acid with sodium citrate, and we dissolve that with deionized water. Then we add sodium borohydrate. That sodium, it normally act as reducing agent, and it change that solution into red color. Normally nano gold will be in red color. Yes, normal nano gold, it will be in red color. So when you add sodium borohydrate, that solution will completely change into gold, that reduce chloric acid into gold, so that will become a red color. When you add titanium oxide and after getting gold, we add titanium oxide in that. We start to stir for 5 hours, and we kept that for overnight, and when you we left that for overnight, and after that we take that precipitate and we washed with we centrifuge and washed with ethanol and water, and we ethanol, and we got this AuTiO2 nanocomposite, that is nothing but gold titanium oxide nanocomposite. And normally here we analyze the formation of Au, that is gold, using this UV spectra. UV spectra is that is optical absorption spectra. First we take spectra of that gold, and it shows strong absorption band at 529 nanometer. Okay, this gold normally it shows a strong absorption band at 529 nanometer, and when you add TiO2 in that, there will be a this absorption band, it shifted to 532. Okay, so it shifted to 532. This plasmon shift, this plasmon band, it shifted, it is red shifted on introduction of TiO2. This normally red shift, it is accompanied when it is accompanied by a suppression of absorption band, it confirms the formation of core shell structure. Core shell structure is center, there will be a one particle will be in center and it is covered by other particle, that we call it as core shell structure. So the suppression of absorption peak and redshift confirms the formation of core cell structure of AuTiO2. Okay, so we also take powder X-ray diffraction of that nanoparticle. So normally this is liquid gold, it is liquid, so we cannot separately take a TiO that XRD, and we just take XRD for TiO2 AuTiO2. So we also we dried this AU particle and we form this as powder and we take XRD, and this is the XRD pattern of AeTiO2 nanocomposite. Okay, this XRD pattern shows both the peak, that is it have both AU peak and TiO2 peak. So it confirms that AU and TiO2 is present in our synthesized sample. This is an FDR spectra, and normally this is due to water molecule present in that molecule, and normally this is due to the metal oxygen bond, and there is some changes in the band from 1000 to 500 centimeter inverse. This is due to the formation of AuTiO2 nanocomposite. Okay, next we here we go for tunneling electron microscopy, high resolution tunneling electron microscopy, that is taken to study the morphology of our nanoparticle. It is just a tunneling morphology of gold nanoparticle we synthesized. Okay, it shows a spherical shape. Okay, normally this is spherical shape, and this is a pattern. Normally using this fringes we can find the lattice distance between the atom. Okay, so this is the d spacing. D equal to 0.2 nanometer. Okay, this EDAX pattern also confirms the formation of gold. Then we add gold with TiO2. From this we can see here it is TiO2 and it is covered. This is gold and it is covered with TiO2 particle. So normally here we can see the interaction. Okay, normally this show. It is covered with a TiO2 gold particle. It is covered with TiO2, and so this is the morphology of AuTiO2 nanocomposite. Next, it is very important study, that is our study. So we are taking here we take a Raman spectra. Okay, Raman spectra, and so see here, this is Raman spectra of AuTiO2. Okay, here we take our lake cell, that is Kodaikanal lake cell. We are finding whether there is a pollutant in our soil or not. Okay, for that normally this is a Raman spectra, and this is normal Raman spectra, that we are take one normal Raman spectra of our soil. Okay, it doesn't show any peak, so we are very happy there is no pollutant. But when we go for SCRS, that is surface enhanced Raman spectroscopy, that here for that we check AuTiO2. Okay, AuTiO2 and substrate, and we coat our pollutant or we coat our soil in that, and we take a Raman spectra. Okay, so it shows continuous peak. This peaks all leads to this type of bands. Okay, this is so this is a traditional technique, GCMS of that lake soil. It shows some pollutant here, so but it shows there is a pollutant. From GCMS spectrum we can analyze that there is some pollutant in that soil, but only in SCRS technique we can find what are the pollutant present in that. Here you can see there are number of C double bond O, C double bond O. These all leads to the these all stretching or lead to the organic pollutant present in that soil. Okay, normally so this can easily identified, the pollutant can be easily detected using this SC or a spectrum. Okay, so first, then here we find the pollutants are present in that compound. Okay, normally organic pollutant are present in that soil. So now we want to degrade that pollutant. For that we use same TiO2. Okay, as usual we take UV spectrum, and from that UV spectrum we plot a graph. Okay, from that we can find the rate of decomposition. Okay, so this is for AU TiO2 and this is for TiO2 with the soil. So this AU TiO2, it shows a higher rate of decomposition. Okay, so it can easily decompose that organic pollutant. Okay, so this AU TiO2 as a nano catalyst, it can easily degrade that pollutant. We also find the stability of that of our compound. So we first measured in day 1 and then day 2 and day 3, all the rate of decomposition was same. So it confirms that AuTiO2 compound which we synthesized was very stable, for the very stable. Okay, so after the degradation we again take the HRTM image for AuTiO2 compound which we synthesize. This is after degradation. Okay, when we add when we add that AuTiO2 with the soil, then it start to degrade that organic pollutant. After degrading, we again take the temperature for that AuTiO2. It was stable as like before degradation. So we can confirm this nanoparticle AuTiO2, it is stable even after degradation. So this is also the efficiency under visible light radiation. So here you can see TiO2, it degrade our organic pollutant, that is 55% of pollutant can be degraded by TiO2, and nearly 70% can be degraded using TiO2 nanoparticle. So this is the another important study, that using our gold titanium oxide nanoparticle we can easily detect the pollutant in the soil, and we can also degrade that pollutant using same AuTiO2. Okay, so normally here that AU and TiO2, it forms as a core-shell structure, and it is easily it provide a significant enhancement of SCRS signal, making it is possible to detect even low concentration of organic pollutant. It shows higher stability and enhanced photocatalytic efficiency. Normally in visible light we found there is only 63%. That graph I miss here. In UV light we can even degrade up to 85%. Okay, so normally this can be used for soil and water treatment. Okay, and next is we like to study about leachate. Leachate is nothing but a brown liquid that can be taken from normally leachate, it is a brown liquid that is taken from our dump site. Okay, so we take normally this landfilling and dumping of organic pollutant and other persistent hazardous compound can be significantly adverse environmental consequence. So that will create leachate. Okay, normally leachate generation is a major problem for municipal solid waste water. Normally our municipality, it will take all the dust particle, all the pollutant from place, and it dump in dump site, and after some days there will be some liquid, unpleasant liquid, brown color liquid. That liquid we call it as leachate. We take that leachate and we try to identify what are the compound or what are the pollutant present in that leachate, and we also try to degrade that pollutant. Okay, so that is that is our next discussion, and this work was published in the Journal of Molecular Liquids. So for that we take, this is normally landfill dust, and from here we collect our leachate. Okay, collect brown liquid. That, see here, this is the leachate. It is a brown liquid with unpleasant smell. This we collect, and we try to identify what are the pollutant present in that, and we also try to degrade that pollutant. Okay, using a silver iron oxide nanoparticle. Okay, so normally has like gold. Here silver also prepared by chemical reduction method. That is, we take silver nitrate and sodium citrate, and we add that with the distilled water, and we add sodium hydroxide. When we add sodium hydroxide, normally sodium boroxide, it normally act as a reducing agent. It will reduce silver nitrate into silver. So in that silver nitrate, we add this iron oxide. This iron oxide is prepared from this ferric chloride and ferrous chloride with ammonia and water. So that gives to iron oxide. Okay, that this iron oxide, it again functionalized with this AU nanoparticle and CTAP. CTAP is a functionalization. So using this chemicals, we functionalize Fe3O4 nanoparticle, and we prepared Fe3, that is silver iron oxide nanoparticle. Okay, so first we confirm the formation of silver and silver Fe3O4 using this absorption spectra. Normally we take UV. This absorption spectra, so this it shows a plasmonic peak. When you add FeO3 iron oxide in this, it will there will be reduce in the plasmonic peak. So it confirms the Ag is added into the iron oxide, and this is the XOD pattern of Ag with Fe3O4. It shows both the peak, Ag peak and Fe3O4 peak. So this confirms the formation of silver with iron oxide. Okay, so next is TEM image of the TEM image of AgFe3O4 nanoparticle. So here there is a dark particle. This is normally due to the silver, and this less contrast particle is due to Fe3O4. So next, this is
Participants, kindly mute your
Sorry
mic.
to interrupt you, ma'am.
Yes, ma'am.
Okay.
Sorry, ma'am.
Okay.
Yes.
Okay. This is an important study. Here we take a leachate. This is brown liquid from the dump site. We take, and we take here, this is a normal Raman spectra of that leachate. It shows only one peak. Okay, then we add silver and FeO3, and we take we take a Raman spectra for that leachate. It shows number of peaks. See here, it consists of number of peaks, and this is due and from this SARS we can find there is a phenol compound in that leachate. That is, chlorophenol is present, cresol is present, phthalate is present, pesticide also present in that leachate, and some heavy metals also present in that. Okay, chromium is present in that leachate. So all this pollutant, phenol, phthalate, pesticide, heavy metals, chromium, it is very hazardous to human health, to human beings. So all this pollutant are present in that leachate. We can identify all that pollutant using this SCR spectrum. Okay, we did that, and next this is for different area. This is G1 is that is 25 kilometer away from the dump site. We collect leachate from 25 meter away from that leachate, and this G2 is nothing but 50 meter away from that leachate, and G3 is 75 meter away from that leachate. All this consists of pollutant. Okay, so we add next we try to degrade that pollutant. For that we go for photocatalytic study. First we take for 0 minutes is nothing but a dye. And when you add your pollute, this is leachate, and when you add our this silver, this is when you add nanoparticle in this leachate, it start to degrade. Okay, it start to degrade. See here, it start to degrade. Uh, so this is the vibrational assignment we already saw. From that UV graph, we can find the rate of absorption. So it is due to direct catalysis and due to photocatalysis. Photo means we use light irradiation to degrade that pollutant, and here we just add that compound. Okay, just add the compound and we find the degradation. So this light irradiation, that is photocatalysis process, have more rate of decomposition. Okay, have more rate of decomposition. So from this study we can summarize that SCRS detection of groundwater represent that leaching of persistent organic pollutant take place in homeland aquifers from landfill. Moreover, there were no vibrational modes of heavy metal CO observed in groundwater. Comparative detection of 3 homeland aquifers enhanced intense Raman signal were obtained from G1. G1 is nothing but a water leachate collected nearer to the dump site, that is from 25 meter. So photocatalytic activity suggests that this silver modified iron oxide or good highly efficient photocatalyst that can degrade the pollutant and retain its optical activity, denoting it can be reused for further reduction activity. So this is our new work, and this is very important work that we already saw. Both Ag and silver, it have more photocatalytic activity. When we add Ag and TO also, we had more photocatalytic activity. So increase the that it have only 83%. So increase that activity. We again add graphene oxide in that compound, and we studied and we try to detect and degrade the pollutant. Here we take just an our dye, aromatic dye, different kinds of dye, and study deduction and degradation of that dye using this graphene oxide nanocomposite. Okay, this paper was published in Applied Surface Science. So for that, first we prepare graphene oxide. Graphene oxide was prepared using hummus method. For that we take graphite flax, and we add sodium nitrate in that, and then sulfuric acid also add in that mixture. We take that mixture in ice bath and we continuously stir that by adding potassium permanganate. Okay, so finally we add hydrogen peroxide and we get on precipitate, and that was washed and centrifuge, that gives graphene oxide again. So then we know titanium isopropoxide with Ag we can prepared by chemical reduction method, and graphene oxide also prepared by Hummer's method. All three was added together, and we transfer all the mixture into Teflon coated autoclave, and we maintained that at 180 degrees Celsius for 6 hours, and we prepare TAG nanoparticle. TAG is nothing but titanium oxide silver graphene oxide nanoparticle. Okay, so that we prepared. So next is UV spectrum of that. It shows this is a titanium oxide. It shows a peak at here. When you add silver, then this peak will shifted to red shift, and again by so this is Ag with titanium oxide. Again we add graphene oxide here and we check that. Okay, so this redshift is due to localized surface plasmon resonance, and we find using this absorption, we using this formula we calculate the band gap. Okay, using this Choff plot. So normally this TiO2 it shows band gap at 3.18 electron volt, and silver with titanium oxide shows band gap at 2.92 electron volt, and silver, titanium oxide and graphene oxide, again the band gap becomes very narrow, and the band gap of that is equal to 2.92 electron volt. Okay, that is 2.65 electron volt. This may due to graphene oxide, which normally graphene, it will be a tubular structure or lamellar structure that contains unpaired electron, unpaired pi electron that can be bond the free electron at the surface of titanium oxide. So it results in reduction of band gap. Okay. Next is XRD pattern. Normally you show here, this is a titanium oxide XRD pattern, and this is with the silver. This is titanium oxide peak with the silver peak, and again we asked graphene oxide here. When you add graphene oxide, the crystallinity it will become reduced, but the graphene oxide will be added in that, and its property also increased. Okay, so this is FTR spectra. So there is FTR spectra also shows there was a change in the band, shift in the band. So it confirms the formation of titanium oxide, silver, graphene oxide. This is interesting. You see here, this is the titanium oxide and silver image, and you see here normally here there you can see a lamellar structure here. Okay, so this see, this is this is silver. Normally it will be in spherical shape, and when you add titanium oxide it will be like this. Okay, group. And when you add graphene oxide, see, this is a lamellar structure, it will be added here. This is silver and this is graphene oxide, and this will be titanium oxide. So from this we can also find the D spacing, this is the fringes. Like by using this fringes we can find the D spacing, lattice spacing of that. Okay, so this is a Raman spectra of titanium oxide and with the silver, and then a titanium oxide with graphene. This is a important peak of graphene oxide. This is due to scattering of Raman. It is a special unique feature of the graphene oxide. We have this two D and G peak when we take a Raman spectra for graphene oxide. Okay, so we studied this. So we study a Raman spectra for this, and then for that we take two eyes, that is we take here 2I, that is Congo red and crystal violet. This is due to Congo red, and this is due to crystal violet dye. And normally this both dyes or cathodic and anionic dye mostly used in textile industry, and it is very dangerous to human health. So we want to degrade that. Before that we want to detect what are the compound present in that. So for that we take a SER spectrum. So it shows number of peaks. For cathode ray, for Congo red and crystal violet peaks are aware. We can identify using this Raman peak, and normally this due to when you add our TAG, that is titanium oxide, silver, graphene oxide, what will happen means normally here this is a graphene oxide. It is helical structure like this, and this is silver, and normally this is titanium oxide. When this is a titanium oxide, normally this is a semiconductor, we have a band gap on that. Okay, so it normally gives electron here, and silver, what will happen means this silver will placed in front of this graphene oxide. So this spot we call it as hot spot. In this hot spot, that is the nearest space between junction between these two particles, we call it as hot spot, that is graphene oxide and silver and titanium oxide. This we call it as hot spot. In this hot spot there will be enhancement in electromagnetic field. Okay, so it will give more Raman signal here. So because of this the Raman signal get increased, or it will get enhanced, and we can easily identify the organic compound. Even even single molecule can be identified using this. Okay, next we want to degrade. For that also we take the two dye, congorate and crystal violet dye, and we start to degrade using this compound, that is titanium oxide, silver, graphene. Using the nanocomposite we degrade and we find the rate of decomposition. So this TGSO more degradation than silver and silver titanium oxide. It also, we also measured degradation for 1 week. That is, we measured first day and third day and seventh day. All three have same rate of decomposition. So it stated that the compound is very stable even after 1 week. Okay, so this normally takes place when you add a dye with a dye, when the light is irradiated with the dye particle, the dye get excited. So when it react with the titanium oxide means it will produce electron, and when silver irradiated with light means it will produce both holes and silver. So this act with graphene oxide, and this graphene oxide will interact with the dye molecule and it produce OH, that is hydroxyl. It is very degradable product. This dye is undegradable, and this OH product, final product OH, it is degradable product. So we can easily degrade that. Okay, so this what happen when you add our nanoparticle in dye, that get easily degrade. So the wastewater with dye can easily degrade using this nanoparticle. Okay, normally this will takes place. Okay, this is a graphene oxide and the titanium oxide. We have a band gap here. When silver, when it is react with this, it will produce electron, and when light is fall on it, it will produce electron and hole. So because of that, it will produce OH particle, and again the graphene also takes it will produce an electron, and it reacts with the dye product. So it will when reacted it with dye, it will produce OH. That OH hydroxyl group can be easily degrade using this nanoparticle. Okay, that's what takes place here. So this is this is the summary of lost work. That is synthesis graphene oxide TAG with the help of solvothermal method, and we also find the crystalline size using TEM image. We also calculated energy band gap, and due to reduced particle size and reduced band gap, and then it can easily produce surface so it can easily degrade the dye. And when for deduction also, electromagnetic enhancement takes place, and we can easily deduct the organic pollutant using this SERS technique. Okay, that's all about this work. So because of GO graphene oxide, it become highly active, and even in visible light we can degrade the pollutant. Okay, so this nanoparticle, it can be used in industries for sensors and also as a photocatalyst. Okay, so this can be commercialized also. So that's all. If you have any doubt, you can ask me.
Thank you for giving us a very informative lecture on nanomaterials for environmental remediation, which will be very useful for us to pursue research in future. Now the session is open for queries. Participants can unmute your mic and ask your queries to the chief guest.
First of all, I would like to thank our chief guest, Dr. R. Parimala Devi ma'am, for accepting our invitation and being here with us today to deliver a talk on nanomaterials for environmental remediation. You have talked about need for environmental remediation and how to detect and degrade soil pollution and water pollution by nanomaterials, and it's very much informative, ma'am. Once again, thank you, ma'am. Next, I thank our management, our department, and our principal for this wonderful opportunity. On behalf of Department of Physics and Chemistry, I thank all the participants for making this webinar a grand success. Feedback link will be posted in chat box and WhatsApp group. Participants can give your feedback. Once again, I thank one and all present here. Thank you.